Um, so, hello everyone. My name is Rubén Chazarra Gil. I'm a PhD student at the Transcriptomics Labs at the Barcelona Supercomputing Center. I'd like to thank the organizers for giving me the opportunity to present in this first LGBTQ plus symposium. And I'm going to be talking about my latest PhD work regarding alternative splicing variability between human populations using single cell data. So, in my lab, we're interested in understanding inter-individual variation between humans, and to do so, we choose as molecular layer the transcriptome. In particular, the type of variation I am studying in my PhD is that one arising from individuals being from different geographical locations or from different genetic backgrounds. This type of variation is a variation that has been traditionally neglected in science, which has mainly focused in white male individuals of European ancestry. And you can, for instance, see this in the total fraction of the GWAS studies, for instance, that include the different ancestries, with the majority being European. So while this different genetic background differences in gene expression at the gene expression level have been widely characterized, we lack a comprehensive characterization of alternative splicing differences between different populations, because such studies have mainly focused on heterogeneous tissues through bulk analysis sequencing, or on single cell types that have been isolated by FACS, for instance. So our goal really, or the goal I'm pursuing in my PhD, is to understand inter-individual alternative splicing differences at the cell type resolution using single-cell RNA-seq data. And to do this, the dataset that we are using is a dataset composed of 90 individuals of mixed European and African ancestry, published by the Barreto Lab in 2021, from which blood and peripheral blood mononuclear cell fraction was extracted, and then those cells were cultured under a non-stimulated condition and influenza and under influenza A infection. After all the preprocessing, this leads to a set of 220,000 cells in which we can recognize and confidently assign the different expected cell types that we have within PBMCs. So regarding the methods that we are using to characterize alternative splicing, the first thing that we have to do is quantify transcripts, or features, better said. And for this, we are using Callisto-Bustos method, which is a very fast method that is characterized by not outputting a single transcript quantification, but instead what is called transcript compatibility counts. Really, what the method is doing is checking the compatibility of k-mers of the query reads with k-mers of the reference transcriptome, and then outputting a table of features which may correspond to a single transcript or to more than one. We also have to mention that since we use 10x data, in which we sequence just a few bases from the 3' end, we are expected to capture those alternative splicing events that show variability in the 3' end. So regarding the numbers that we can detect, these are the number of transfer compatibility counts with a corresponding number of genes, and we can see how at the individual cell level we're able to detect few hundred TCCs corresponding also from a few hundred genes. We can highlight monocells where we have a higher transcript diversity in this case, and this novel gray numbers justify the need to pseudobulk, which is basically aggregating the different counts per cell type and per donor, in which case we are able to detect many more transcripts also corresponding from more genes. The next step that we're conducting is differential transcript usage, which basically is modeling the relative abundance of the transcripts within a gene across the trait of interest, which in our case is the genetic ancestry. So basically, on a per cell type and per gene basis, we're modeling the relative abundances of the transcripts of a gene as a function of technical covariates, biological covariates, and genetic ancestry. Interestingly, we are using a continuous estimate of genetic ancestry because we have the genotypes of those individuals, so we can avoid doing discrete variable and using a continuous genetic ancestry. To do this modeling, we're using MANTA, which is a recently published method from which we leverage the fact that it's very fast and not parametric, so it does not really assume a particular distribution of the fractions of the transcripts, which is very convenient using single cell data. So what we find, we basically find few but some genes that change the relative abundances of the transcripts across the different immune cell types of the blood. We also notice that the signal is very correlated with the cell type size, with the highest number corresponding to the cell type that is most abundant. And also, depending in the characterization that we are doing, we see that for the majority of genes that we test, we have two features, two transcript compatibility counts that we model, and for some of them we have an increasing number. Also, we find interesting to mention that the signal that we capture with this differential transcript usage is partially independent from differential gene expression, and this is interesting because we would expect that upon an overall increase or decrease of the gene expression between populations, there could be stochastic shifts in the transcript ratios, but actually what we see is that this signal is quite independent from an overall increase or decrease in gene expression. As an interesting example of gene that is population differentially transcript used, we can highlight immunoglobulin heavy constant alpha 2 gene, which corresponds, or which encodes for the structure, the highlighted structure of immunoglobulin A, which is a very abundant antibody in our body. And what we'll see here is basically the transcript models of transcript 1 and 2. As you can see, these differ in the 3' end, which is represented in this sense, and this gene is characterized by 3 different equivalence classes from which we get those transcript compatibility counts, which corresponds to a unique region in transcript 201, a unique region in the 3' end of transcript 202, and then a region which is common to both. We can already see through the coverage lines, which are here highlighted for African individuals and European ancestry individuals, that the yellow line corresponding to African ancestry individuals spans through the unique region of transcript 202 and also reaches about the unique structure of transcript 201. This, once we visualize the raw data, we can see that while for African individuals there are 2 different transcripts expressed, the first one and second, for European ones, we mainly find expression of transcript 202. Here we can see the same, but showing the relative abundances of the different features, where each column corresponds to a single donor. And this is also interesting. I wanted to highlight because population diversity has been seen in this family of genes at the genomic level, and it's interesting that now we can report this at the transcriptomic level, at the expression level. Next question that we asked was if there were differences in the relative abundances between infection status. This is between flu-infected samples and non-infected samples. And here we model it a bit differently due to the fact that the data is paired from the same individuals. And here we are interested in the intercept, which is basically the coefficient telling us if the subtraction of the flu relative frequencies and the non-infected relative frequencies is different from 0. And in this case, we are not that lucky finding infection DTU hits. We find very few, but at least once visualizing the ones that we're able to retrieve, we do believe the signal. And again, we're showing a gene, in this case, which is a protein phosphatase, which also differs between those transcripts in the 3' end. And we can see while some of the features barely differ from 0, the subtraction of the relative frequencies of flu and uninfected, some of them don't differ, some others increase upon flu, and some are, some of them decrease with flu, while others increase. Next thing that we were interested in is inferring, or how could we infer potential functionality of the alternative splicing differences that we find. And for this, we developed an in-house pipeline in which we're basically extracting the coding sequences of the transcripts and then mapping those to the protein domain database PFAM. Okay, here you can see as a as a quick example, gene again with two different transcripts. Here in gray, I'm representing the different domains that were mapped to those coding sequences. And you can see that while for some of the domains, like this ALD domain, here this P equal 1 represents that the full domain annotated in the database is present in the coding sequence. For some of them, in this case, 78%, is only present in that coding sequence. So this serves as a proxy to infer potential functionalities of the alternative splicing events that we are able to capture. Finally, as some validations, we carry on permutation analysis in which we permit the labels, in this case of our population, DTU analysis. Here in green, we can see the number of DTU genes that we report with the correct labels, and here in this box plot at 0, we can see the number of DTU genes that we find when we shuffle the labels, and this was across 10 iterations. And we additionally perform an overlap with the DTU signal that we get from bulk RNA-seq data from matched samples, and as you can see, we will report quite a high overlap for the main cell types. So as a conclusion, I wanted to highlight that inter-individual alternative splicing characterization is now possible thanks to the fact that we have population-scale single-cell RNA-seq datasets. We present a solid strategy to perform differential transcript usage with 3' single-cell RNA-seq data, which is challenging. And we find some alternative splicing events across blood immune cell types, which are partially independent from gene expression, and some of which present some potential implications. And before, well, I would like to thank all my team, especially my PI, Marta and Ida, who's helped me a lot with this analysis. And before finishing, I'd like to highlight an initiative that we're carrying on at BSU. This is our research center where we proudly host Mare Nostrum 5 supercomputer that has been recently inaugurated. But what I really wanted to comment is about this initiative that we started sort of like a year ago, which is the BSC Queer Group. It basically started by informal conversations in which we noticed that while other research centers in Europe, where some of us had been, did have an active queer visibility group, let's say, we were lacking such a structure in a research center which has more than 1,000 employees. So we started this journey in which, this is a bit of a timeline of what we did. Last year, we started in March 2023 with the Trans Visibility Day, where we could present the group, and it was very successful. A lot of people came, and since then, we've been conducting activities, for instance, for the Lesbian Visibility Day, for the Pride, for Bisexuality Visibility Day, also for Asexuality, and inviting queer people to give some seminars. Science-related or not science-related, to be able to host them in our space. And this is something which we're very proud of, which is having a lot of success, and it's quite cool to see how many people from many different identities of the community have approached us and thanked us for this initiative. And it's also good to see that it comes from the workers, so bottom up. And with this, I'd like to thank you all for your attention. I'm happy to take any questions.
Thank you, Rubén. Any question from the audience? Rubén? One short one. I have a big one. The question is me. Do you think you can adapt your pipeline to use SmartSig3 data, maybe discriminate between different platforms in your analysis?
So, very good question. Our main goal really in the lab is checking inter-individual variation, because some people come to me and say, if you want to look at alternative splicing, why don't you rely on SmartSig, which covers the full transcript, so you can detect more alternative splicing events that have variability in the 5' end, for instance. But truth is that such population scale data sets where we have hundreds or even thousands of individuals are available for 10x mainly for a reason of cost. So focusing on variation between individuals, we now have to rely on 10x. I'm aware that when long-read technologies scale at the single cell level to population to population cohorts, that will be more powerful to detect such changes. But at the moment, is what we have, and we still find some stuff. So it's good to see that.